Hi everybody, and welcome back to Geology 109, Evolution of the Earth. This is week 7 of the spring 2021 semester. We're going to continue our trek through geologic time. Last lecture series, or last week I should say, we discussed plate tectonics and kind of the evolution of the Earth in that way. But this week we're going to actually look at Precambrian history. This is what's happening kind of as plate tectonics has now been established. Really, we're looking at a vast, significant majority of Earth history in one lecture, or one lecture series. So this is going to be covering 4.6 billion years to about 600 million years of Earth history. Again, a vast majority of our planet's history. And also part of this involves the earliest forms of life. So let's jump right in and talk about the Precambrian. Now, most of the time when we look at a complete geologic time scale of Earth's history, it looks kind of like this, where a majority of our Earth history is actually being shown really over the last 540 million years, and everything past that is this little sliver down at the bottom. But if we extrapolate this out to its true representation of time, slightly more to scale, you can see that the Precambrian makes up an incredibly massive portion of Earth history. For some reason, though, mostly because we have a better record of it, we like to focus on the Phanerozoic, which is the last 540 million years. And we're going to get to that after this next exam. But we're going to be focusing this lecture on just the Precambrian and the three subdivisions, the sub-eons of the Precambrian, the Hadean, the Archean, and the Proterozoic, which span collectively from about 4.6 to 4.5 billion years ago to 540 million years ago. Really, when we start getting multi-celled organisms that have hard parts like shells, that's where we draw our boundary to say, now we're in what's called the Phanerozoic, which again means visible life, meaning we now have a visible, easy to recognize fossil record. We have fossils from the Precambrian, but they're not as easy to identify, usually, as what we have in the Phanerozoic. So we're going to talk more about what's going on during this major, significant early portion of Earth history. A majority of our Precambrian record, obviously, is going to be tied up in Precambrian rocks. So where do we find Precambrian rocks? Well, if we focus on North America, you can see where we have what's called the Precambrian Craton. This is the preserved blob, essentially, of Precambrian crust, continental crust, in North America. And it's made up of these smaller provinces, what we call the Ray province, the Nain, the Hearn, the Wyoming, the Superior, and the Slave province, all kind of crushed together. And most of it, yes, is covered by relatively more recent sedimentary rock. And the Precambrian basement rock that's there, which is, as you can see from the map currently underneath where we are in Wisconsin, is highly metamorphosed. In other words, if you could go back to, let's say, the latter portions of the Precambrian, let's say the Proterozoic, if you wanted to look at North America at the time, pretty much what you see labeled Craton, that's what we would have had. Probably not even that much, because a lot of it would have been underwater. So our Precambrian rock record, think of it this way. This is the stable regions, what we call the Cratons, the basement rocks. These are the foundations of all of our modern continents. So what is a shield, what is a platform, and what is a Craton? Well, a shield is a large body of Precambrian crystalline rock, whether igneous or high-grade metamorphic, meaning this could have been volcanic rock that was produced during the Precambrian. It could have been granitic rock that was forming deeper into the crust, but this made up a significant portion of our continental mass in the Precambrian. Now, by today's standards, it's been heavily metamorphosed, especially into things like gneisses. The platform is the sediment that would have piled up around that continental crust back in the Precambrian. So you'll have this large mass of igneous rock sticking out of the sea, and as it erodes, because there's nothing else to do, that sediment's going to pile up around the periphery of that shield. Collectively, we call this a Craton. A Craton is a combination of the shield and the platform. So the basement rock would be your shield. The platform is the sediments that are cascading and eroding off of that shield. And collectively, we call that a craton. So the cratons we talk about in North America that are Precambrian in age are not only the Precambrian basement rock itself, but also some of the Precambrian and later sediment that piles up around it. The key here is that it's tectonically stable, meaning these aren't areas that have undergone relative or geologically recent tectonic activity, uplift, rifting, or transform boundaries. So that's why we look at, especially up in Canada, we actually call that the Canadian Shield, because North America's basement rocks, really our oldest rocks, are up in the Great White North. We are currently on part of the platform. So we're still on part of the craton, but we're on the platform portion of it. So again, just focusing on North America, in the orangish color that you can see, that's the Canadian Shield, or the North American Shield. That's all Precambrian igneous and metamorphic rock. All of the green that you see deposited is part of the platform. So we are currently on part of the platform, but northern Wisconsin is part of the shield. Collectively, we are all on part of this North American craton. Now, what about south and west of that? These are areas that, at least if you look west, what's that kind of bordering? That's the Rocky Mountains. That's an area that's been tectonically active in the relatively recent geologic past. If we look to the south, like around the Gulf Coast area, that's all sediment that's actually been deposited over just the last 65 million years or so. Or if you look further north around New England, that's areas that have also been modified due to the Appalachians. So it has to be tectonically stable throughout a significant portion of Earth history. And that's where we are. We haven't seen a lot of tectonic activity in Wisconsin since, well, the Precambrian. And all of the green, what you see platform, that's all sediment that's deposited off of that Canadian Shield in the Precambrian. So the basement rocks in North Texas, for example, those are platform sediments from the late Precambrian throughout most of the Paleozoic, but they have not been tectonically modified. And while we are focusing on North America, I do want to show you that this isn't unique to North America. There are shields and platforms and thus cratons all around the world. All of our continents are made of a shield, which is the base, earliest, oldest Precambrian rock. There's a platform of sediment that has shed off of that back in the Precambrian and the early Paleozoic era. And collectively, we have these cratons. And anywhere that you see yellow, for example, that's extended crust. Those are areas that have been geologically more recently stretched. The purple are more geologically recent igneous provinces and eruptions. The light bluish greens are mountain building events, so again, not tectonically stable. And the darker, or the bluish patterns are basins that are also in some cases tectonic. So if you want to focus on what actually is the Precambrian rock, you're looking specifically at the orange pattern. And if you want to see what's part of the platforms of those cratons, you're looking at that kind of pinkish color. Collectively, back in the Precambrian, all we would have really had were those shields and some very early sediments that would have been coming off of them. So again, in North America, if we look at just kind of those reddish warm color patterns, that's our Canadian Shield, right? That's our basement Precambrian rock, the oldest rock. That's the igneous and metamorphic rock from the Precambrian that makes up the core of the North American continent. Now, there's some other places where we see these Precambrian rocks exposed, but they're usually places where rocks have been severely uplifted from deeper in the crust. So mountain ranges where we uplift parts of the crust. So the Appalachians, which are very, very old, and the Rocky Mountains, which are considerably younger, do show some uplift of these basement Precambrian rocks as well. So the first 3.8 billion years of Earth history, what we collectively call the Precambrian, is really subdivided into 3 sub-eons. And they are, in order of oldest to youngest, the Hadean, the Archean, and the Proterozoic. Now, we don't really have much for Hadean rocks. This is during a time when there was a lot of meteorite bombardment. The Earth was still forming and cooling. This is when we get our first oceans. We get our first versions of an atmosphere. and the continents are just starting to form. The Archean is a much more kind of defined record, and then the Proterozoic is a fairly decent record. There's just not a ton of exposures. What makes these three subdivisions a little more interesting is life. Even though this is 88% of Earth's history, we actually see that life forms very early. There's a chance that life may have started in the Hadean. We know that it starts in the Archean, and we actually know that it's sticking around and a little bit more prolific, and we start getting our first multicellular organisms in the Proterozoic. So even though the name Proterozoic means hidden life, we know that we have life there. The Archean is when it first appears or at least remains viable. We think that in the Hadean, conditions may have been right for early life, but life may have formed numerous times. Now, those conditions are no longer what we see on Earth today. So we shouldn't expect spontaneous life to be forming anymore because the conditions just aren't right for that anymore. Plus, we already have a lot of life that uses up the materials that we need. And we'll get into more of that in a couple of slides. But I just want to set the stage that from 4.5 to 4.6 to about 3.8 billion years ago, that's the Hadean. From 3.8 to 2.5 billion years ago, that is the Archean. And the Proterozoic is from 2.5 billion years ago to about 540 million years ago. So these are huge spans of time. And while there's not a lot of active, exciting, these aren't action movie periods of time, there's some very, very important events happening that sets the stage for the continued evolution of our planet. But let's talk a little bit more about the Hadean, or what we know about it. Now, this is an artist's rendition of what we think the Hadean may have looked like, where you have some craggy early proto-continents sticking up out of a largely molten surface and a moon that's very, very close and a lot of meteorite bombardment. But let's get into a little bit more about what we know about the Hadean. This is really the first 500 million years of Earth history. So it's an important time when the Earth is going through the last stages of its formation and it's starting to stabilize. In the beginning, the surface was largely molten, and by the end of the Hadean, it had cooled off to actually becoming a crust. There's very few microcontinents that are beginning to form. In other words, we're seeing a differentiation between basaltic ocean crust and granitic continental crust, or granodioritic continental crust. And we wouldn't see large continents at this point. These are kind of more microcontinents, little islands of granite masses sticking out of a mostly basaltic molten surface. There would have been a lot of meteorite bombardment. Remember, the solar system is still very young at this point, and there's a lot of debris still floating around. And our planet, as well as all of the others, are getting constantly hit by meteorites and asteroids. And as this happens, it continues to heat the surface of the Earth. So we have some cooling and then remelting and cooling and remelting with repeated bombardment. And eventually that starts to slow down. Because the moon would have been very, very young, it would have been twice as close to the Earth at the time, and thus had an incredibly strong gravitational pull. Now today, the moon has a gravitational pull on the Earth that affects our tides. So imagine the Hadean, where you have tides of basaltic lava going up and down rather than just water. The early atmosphere of the Hadean Earth would have been incredibly hostile to organisms by today's standards. There would have been a lot of nitrogen, but also methane, ammonia, carbon dioxide, and water vapor, though no free gaseous oxygen. You wouldn't have been able to walk around, well, anywhere in the Hadean and take a nice deep breath and get oxygen. It would have been a very hostile atmosphere for living things by today's standards. And also, in the early Hadean, no oceans. Though, well, maybe. There's some relatively recent discoveries in Australia in some very, very old Precambrian rocks that suggest that perhaps by the end of the Hadean, we're starting to see our earliest forms of oceans. Just a couple of years ago in the Jacks Hills of Australia, some zircon crystals were found that date to about 4.4 billion years ago. Now, zircons are a mineral that form in sedimentary environments. They're one of the few things we can look for in sedimentary rocks and directly use absolute age dating on them. So these zircon crystals date to about 4.4 billion years ago, so in the Hadean, but also it's important to note that zircon crystals form when you can have warm oceans, but it has to be cool enough to form them. So this kind of changed the way that we looked at the Hadean a little bit, that the Hadean Earth may have, by the time we start going into the Archean, we're actually starting to see oceans cool enough to start forming zircons and thus potentially harbor life. But again, I don't want to get too far ahead of myself. We're going to get into what we know about the origins of life, but this lets us know that the stage was set by the end of the Hadean, possibly for that process to begin happening. So let's look at North America during the Archean. Again, if you look at the Canadian Shield and Craton of North America, you'll see that it's made up of chunks of continental crust that have all been kind of slammed together. So most of our surviving Precambrian rocks are mostly granitic in composition, meaning they have potassium feldspar, quartz, some hornblende, and lesser amounts of micas. These are arranged, though, into these highly metamorphosed, what we call orogenic belts. Orogenic belts is another way of saying deformed mountain ranges. So an orogen, O-R-O-G-E-N, is a mountain belt. So we have these ancient mountain belts, and by today's standards, they've been whittled down and covered with lots of sediment. But how we actually produce these involves a system called collage tectonics, or continental accretion hypothesis. If we date these chunks of Precambrian rock, even though they've all been slammed together, we see that the ages vary. We have some that are Archean in age and some that are Proterozoic in age, but most of the ones are Archean, but even then we have some that are early Archean, middle Archean, and late Archean. And if we reconstruct the environments of what these were like, we're seeing that this is when plate tectonics was really an active event. So all of the green that you have here are Archean terrains that have been metamorphosed. So let's look a little bit at what this collage tectonic process looks like. But first, let's start by looking at what kind of rocks we're dealing with. So again, our Archean era, which is about 3.8 to 2.5 billion years ago, are mostly granitic in composition, but today they've been metamorphosed into more gneissic textures. So we have those alternating light and dark bands, but the compositions are definitely granitic. Okay. And these have all been slammed together to produce our larger continents, including the North American Craton. And this seems to stop somewhere around 2.5 billion years ago, around what we call the Proterozoic. That's when we're starting to see the shields and some of the platforms of our larger continents. And it doesn't mean the continents would have looked the way they look today, but their basements were kind of done being assembled. Okay, so continental accretion is the process by which we are slamming little microcontinents together to produce larger continental masses, and most of this is happening during the Archean. And we know about this from these structures called greenstone belts. Okay. Greenstone belts are these bands that are kilometers wide of metamorphosed basalts, so the stuff that makes up the seafloor today, as well as gray wacky sandstones, the type of immature clastic sedimentary rocks that we see associated with erosion and almost immediate deposition. Pillow lavas and turbidity flows, all of these things, the basalts, gray wackies, pillow lavas, and turbidity flows, these are all things we find today making up a significant portion of our ocean floor. So when we find these metamorphosed from the Precambrian, we interpret these as wedges of ancient seafloor. So the idea is our little granitic microcontinents in the Archean would have collided with each other. The ocean floors that are in between them, we get small wedges of those that get pinched and collapsed and metamorphosed together. And that's what we call a greenstone belt. So a greenstone belt is a metamorphosed band of ancient ocean floor made of metamorphosed basalts, gray wackies, pillow lavas, and turbidity flows that represent the ancient seafloor that got wedged in between these small microcontinents or protocontinents in the Archean. So this is the last bit of surviving seafloor from 3.8, 3.5 billion years ago. And they're these wedged in between these gneissic terrains that we see up in Canada. So just to clarify, if you go up to the Canadian Shield today and you do a geological survey, you're going to find metamorphosed granites in the form of gneisses, and you're going to find in between them these very large bands of greenstone, of this mixture of basalt, meta-sediments, volcanic sediments, and pillow lavas and turbidity flows. And it's this repeating series. If you start dating the ages of the gneisses, which used to be granites, and the greenstone belts, which used to be seafloors, you start finding that the oldest ones are in the center and they get progressively younger outward. That's because you'd collide two continents, and then two more continents, and then two more continents to produce a larger continental mass. So on this diagram, we can see, if we look up here in Hudson Bay, okay, we have our main Precambrian shield, okay, in Canada, and we have these long green bands. Now they're just colored green for this illustration, but this is what, at the lower part of this diagram, this is what they actually look like today. In cross section, we have a lot of the greenstone belts. They're being ancient seafloors, ancient volcanic events wedged in together of metamorphosed continents. And if we look at our central diagram, that's an interpretation of what would have been going on back in the Archean. So you had small continents, and they had divergence happening in between them, making them even smaller, and then subduction happening at the opposite ends, and then the compressive forces of convergence slammed them all together. So what used to be the subduction zone, what used to be the back arc basin, what used to be those volcanic areas are now metamorphosed into greenstone, and they're surrounded by the granites, now metamorphosed into gneisses all around them. So essentially, greenstone belts represent the scar tissue of how our continent was built from colliding one microcontinent into another, and they accreted to be much larger structures over time. So this was a time, the Precambrian was a time when we had a lot of tectonic activity happening in North America. Not just the construction of the shield itself, but divergence being a really important thing. In the previous diagram, you saw that some of those microcontinents were trying to pull together. There's a good example of this, actually, here in the Midwest. Okay. About 1 billion years ago, so much more recently, we had an attempted rift event happening in North America. So still Precambrian, but getting to be relatively more recent. An area that extends all the way from Lake Superior down to southwest portions of Kansas. Essentially, North America almost split into two, and we call this the Quinoan Rift. We know about it not because there's a huge valley or a huge rift section or a big gash or something across the United States, but as North America was starting to rift in two, there was a lot of basaltic eruptions going on, and those basalts, those basaltic lavas, are still preserved today. You see this in places also like up in Minnesota, again, the Lake Superior region, an area called Gooseberry Falls, which is literally thousands of feet of basalt eruption. So you can see in the diagram here, this is the mid-continent gravity high. We call it that because the density of the rock there is much higher than surrounding it because of all of the basalt. So this is what we call a failed rift arm. Essentially, the continent was starting to rift apart, and it stopped for whatever reason. So it's a failed rift, which is probably a good thing by today's standards. But during that time, we were producing a lot of basaltic eruptions, which of course is more dense than the surrounding rock, which is mostly granitic. So we call this the Quinoan stage. It's about 1 billion years old. And if you were to do a gravity anomaly survey over this area where you test the density of the rocks, you find this belt going down all the way to Kansas of a considerably higher amount of density, so thus basalts. And there's places obviously in northwestern Wisconsin where you can see them. It's basalts occasionally some volcanic sediments, but a lot of this is basalts and gabbros. And this is similar to what we actually see happening with the rift system in East Africa today, which we mentioned before when we were introducing plate tectonics. So the rift valleys that we see along Kenya and Tanzania today are analogous to what was happening back in North America during that time. Now, why did the mid-continent gravity high stop? I don't know. This happened. Same reason why the East African rift system stopped. Rifts can start and they can stop, and then they can start back up again sometimes. So let's talk a little bit about what types of sediments we have associated with the Precambrian. If we go to the Archean, so remember 3.8 to 2.5 billion years ago, most of the sedimentary rocks that we see are things that we can classify as greywackies. Now, I want to be clear that this doesn't say every single type of sediment from the Archean is greywacky. Okay. A majority of it is, though. We have a lot of greywackies from the Archean. And again, to remind you, greywackies are immature sandstone. So you start out with something like a granite or a basalt, you weather it and erode it into its sedimentary particles, and they're buried very, very quickly. Okay? They don't have time to refine and get broken down into just pure quartz sandstones. So in greywackies, you will find things like quartz in small amounts, but also other things like hornblende, biotite, pyroxene, feldspars. And normally, with other types of sandstones, the more these are recycled and re-eroded, those other minerals would break down and you're left with just quartz. But in the case of greywacky, they've been buried almost immediately. So as the Archean crust is eroding, sediments are depositing right away, and we have a lot of greywackies. And this makes sense. We have small continents in the Archean. This is while collage tectonics is happening. We have small microcontinents, and as the sediments are shedding off of those, they're piling up around the sides of those really small islands, those protocontinents. They don't have a huge, vast continental shelf to become recycled and reused and broken down into those pure quartz sandstones. So they break off and they get buried shortly afterward. Okay. Now, if you take a greywacky and you do re-erode it and rework it, it will re-weather and you will start breaking down those mafic minerals, those feldspars, the pyroxenes and biotites and hornblendes. And then you'll be left with a more quartz feldspar rich body of sediment by the time we get into the Proterozoic. So the Proterozoic is more associated with these mature sediments, partly because the continents had grown large enough by that time that if you weather a piece of sand or weather a granite or weather a basalt from the middle of a Proterozoic continent, which is much larger, by the time those grains make it to the coast to get deposited, all those mafic accessory minerals have been broken down. So this means that our Archean crusts and rocks and sediments represent unstable tectonic settings. We have rapid sedimentation, not just in the rate of sedimentation, but from erosion to sedimentation, there's a very small window. This is common in areas that have a lot of active tectonic motion going on. Okay, so our greywackies, we have these texturally and compositionally immature sediments. The Proterozoic represents a time of a lot more stable tectonics. We had larger continents. There's still tectonic activity happening today, so we're not saying it's stopped, but it's slowed down. We see more quartz sandstones, well-sorted, more mature sediments. It's almost like if this were an SAT question, it would be Archean is to greywacky as Proterozoic is to quartz sandstone. Okay. This helps us also indicate that we had these larger cratons by this time. So the Archean is a time of constructing continents. The Proterozoic is the time of constructing cratons. We have large shields by the time we start the Proterozoic, and at this point now we're piling up and building up our reworked platform portion of the craton. So this is a diagram from your textbook kind of showing how this breakdown process happens going from greywacky to quartz sandstone. So in the upper portion, we have freshly weathered soil. Okay, so bits of broken rock and mineral. The clays are going to erode away first, or they wash away first. So we're left with more coarse grained material. And then through fluvial or river transport, we start sorting things out. The silts go in one place, the pebbles get left behind, and we're left with just more of a raw quartz body. These grains become rounded and concentrated as a sand. Now we can also do the same thing with a parent rock like granite. In this case, we have a rock that's made of quartz, potassium feldspar, biotite, and as that chemically weathers, those feldspars and quartzes and biotites start to break down. The calcium, sodium, and magnesium ions break down in solution, and so now we have rubble. We have broken little mineral bits from this rock that's broken down. Some of those feldspars will break down, and biotites and amphiboles will break down into clays and will be washed away. Now, at this first stage right here, which I'll circle, that's where we would stop at the gray-wacky stage. So that's predominantly what we are depositing in the Archean. By the time we get to the Proterozoic, though, we've moved on to the next couple of stages. And as you keep winnowing these things out more and more, eventually you're left with just a quartz sand residue, but it's abundant enough to make up vast amounts of quartz sandstone. So this is what's happening more in the Proterozoic. We're stopping at our first stage here in the Archean. So that's why we say the Archean, we see a lot more gray wackies, and in the Proterozoic, we see a lot more quartz sandstones. Now, here's a photomicrograph of a gray wacky. So again, we took a gray wacky, we cut it and polished it and made it into a thin section that we can look at under the microscope. And as you can see, this is a first cycle rock. These are grains that broke off of a granite or a basalt, and they were pretty quickly deposited and stayed that way. We have large grains, small grains, a whole mixture of different elements. This is what you would have seen on the beaches in the Archean. These are the types of sediments that would have been piling up. So compare and contrast that with something like a quartz sandstone. Now the different colors, the shades you're seeing here, the black, white to gray, that's actually just the crystallographic axes of those individual quartz grains as light's moving through them. You're actually looking here at about 90% quartz. Notice the grains are all rounded. They're roughly the same size and shape. This would be some second, third, or nth cycle. This is what happens when you take a gray wacky or raw weathered material from rocks and you recycle them over and over and over. And you need a large craton to do this. We don't start seeing a lot of quartz sandstones until the Proterozoic. We still see our gray wackies today. We do still see gray wackies in the Proterozoic, but they're not as abundant as a percentage as what we saw back in the Archean. So we're talking about sediments, and we've talked about how we produce continents, but how does this play in with our oceans and atmosphere? And this is where the Precambrian really starts making some major changes. If we go back to the early Archean, the atmosphere was what we call anaerobic, meaning it had no free oxygen. Remember, there's oxygen on the planet, but it's tied up in other rocks and compounds. We don't have gaseous oxygen in the atmosphere. So if you went back to the early Archean and took a deep breath, you'd suffocate. There was no oxygen. But it was pretty rich in carbon dioxide. How do we know that there's no oxygen in the Precambrian, in that early part of the Precambrian? Well, because we look at some of the minerals and rocks that we have preserved from the Archean and find that these are things that today would oxidize or rust. So, if you take a piece of metal and you stick it outside and it gets rained on, exposed there, it'll start to tarnish and ultimately rust. We live in the Midwest. This happens to all of our cars, right? When we look at our Archean rocks, we find things like carbons and iron sulfides and iron carbonates, and none of them are oxidized. Today, they would be oxidized as they're forming. So if you have unrusted rocks that should be rusted, that indicates that you had no oxygen in your atmosphere. Any little amount of gaseous oxygen that would have escaped maybe from the weathering of a rock is going to get used up by what we call oxygen scavengers, like hydrogen and carbon dioxide. We'll use that to bond, like water vapor and CO2. So we don't have a surplus of oxygen. Again, you couldn't go back in time and take a deep breath. Okay. Once we started building up enough oxygen, then we start to see sulfurs and irons and manganese start to oxidize more. But we see a very, very slow buildup of oxygen initially. And actually, we don't have much for quite a while. It's about 2 billion years ago, out of 4.6 of Earth history, 2.6 billion years ago, we actually get oxygen to about half of the concentration just today. And if you remember from class today, oxygen makes up about 20 to 21% of our atmosphere. So it still wouldn't have been very easy to breathe even 2 billion years ago. But then something interesting starts happening. In the late Archean, early Proterozoic, we start to see a new type of sedimentary rock forming in very high abundance around the world, and these are called banded iron formations. And as you can see in the image, they're these alternating bands of purple and pink rocks. Essentially, it's a mixture of hematite and iron oxidized rich chert. The reason that they band back and forth is due to some pH issues. So there's chemical changes going on in the ocean, which is changing the pH, and as a result, we're starting to see the buildup of oxidized iron, essentially hematite. It's rust, that's iron oxide. We start to suddenly see this, and this indicates that somehow we're getting a larger amount of oxygen in the atmosphere, because that's allowing the water to oxidize and the minerals dissolved in that water to oxidize and precipitate out as banded iron formation. So we start seeing banded iron formations, which are these interlayered chert and iron oxide layers. About 2.5 billion years ago, they start becoming very, very abundant. So right around the beginning of the Proterozoic. What does this mean? It means that oxygen concentrations in our atmosphere had started to reach high enough levels to be a surplus. You had enough to oxidize the irons and sulfides, those little oxygen scavengers, the CO2 and the hydrogen, but you also have enough oxygen then to start oxidizing and weathering other things. We have a surplus of oxygen suddenly about 2.5 billion years ago. Now, if you ever drink water out of a, Wisconsin's call them bubblers, I'm from Illinois, they're fountains, drink out of one of those and you taste metal, tastes kind of rusty. That's iron oxide dissolved in the water. It doesn't hurt you, it just tastes kind of weird. Because there's two different types of iron ions. There's Fe2+ and Fe3+. Fe2+ is commonly dissolved in seawater. When it oxidizes, it gets that funny taste, and if it gets in a high enough concentration, it will precipitate out of the water as a solid. So when you oxidize that Fe2, it'll convert to Fe3 and then start to precipitate out. And that's what we're seeing happening with banded iron formations. So the iron 3 starts to form in the water as a solid and fall out of suspension and starts piling up on the ocean floor. Now that's all fine and good, but where did we get all of this free oxygen suddenly? Where did this O2 gas in our atmosphere suddenly come from? Well, that is one of the first indicators, not the first, but one of the first indicators of early life. That the oxygen in our atmosphere, the first oxygenation events in our atmosphere, were due to organisms. Simple, single-celled organisms that were now capable of proliferating because they had continental shelves that they could start building up colonies on and producing enough oxygen to oxygenate not only the surface waters, but also the atmosphere. And so I want to shift gears a little bit and talk about what we know about the origins of life. And I'll start with a strong caveat that no, we do not have all of the answers. Scientists have not figured out all of the answers regarding how life started. However, we have a significant portion of the puzzle that kind of points in one direction and away from others. And as the great astrophysicist Carl Sagan once said, these are just some of the things hydrogen atoms do given 15 billion years of cosmic evolution. But what we're going to be looking at now is kind of what it takes to form life. What kind of conditions would we have needed? What kind of conditions do we know we had around in the early Earth history? And how life really gets going and how it keeps going then, of course, through evolutionary processes. So remember, if we look back at the early Earth, what are some of the physical conditions that we have in the early Earth's timeline? Now remember, we said if you could go back to the Hadean and take a deep breath, it would not be very hospitable to you. That's by today's standards for life. But remember, there's living things that can breathe things other than oxygen. We happen to be aerobic organisms that breathe oxygen and exhale CO2. Plants, for example, do the opposite. But there's other organisms that can breathe and emit different gases. So if we look back at the Hadean to Archean, the late Hadean, early Archean, around the time we're starting to see those early oceans forming, and they're still warm, but not boiling, the temperatures would have been in the correct range for life. If you can form zircons, you can certainly host living things. We have general water as a fluid, meaning that it's not, we don't have a surface so hot that we can't hold liquid water, and it's not so cold that it's all frozen. We're in that nice, what we call the Goldilocks zone. It's not too hot, not too cold. It's just right. We know that the early atmosphere, again, lacked oxygen. It wasn't until the Proterozoic we started to see that happen. And therefore would have been very reductive. We're going to have oxidation and reduction, and this would have been very reductive. We know that we had a lot of energy bombardment from the sun. The sun was still very new, relatively new compared to today's standards, and we were getting a ton of bombardment of solar radiation. So what exactly is life, though? Well, not to get spiritual or metaphysical, but life is essentially these four main things. Because if you're talking about cats, dogs, birds, humans, or microbes, we really got to look down at what is the most simple form of life, and that would be a microbe, a microorganism, single-celled microorganism, like a bacteria. It's a package of complex organic molecules. That's really what it is. It's a cell that contains complex organic molecules. It is self-regulating, meaning it can keep itself going. It's self-replicating. Earliest forms of life asexually reproduced. They essentially clone themselves. And it has self-propagating chemical reactions. And then we can think about, well, what about deep thought? Yes, that's something that, some other advanced forms of life, like humans, do, but that's still self-propagating chemical reactions. So if you boil down the very basics of life, we're looking at these four things. So this isn't the question of how do you build a human in the Precambrian, because we're not suggesting that. It's how do you get cells? Cells that are made of complex organic molecules that can self-regulate, self-replicate, and self-propagate through chemical reactions. A little bit. So how did life actually start? Well, I'll be the first to tell you we don't know 100%. We really don't. We have not been able, as scientists, to completely replicate the origins of life in a lab. We've gotten very close, but we're talking about a process that initially happened possibly 4, maybe 3.8 billion years ago in a very different planet, or a very different type of planet than what we see today on Earth. And replicating that absolutely perfect set of circumstances is very, very difficult. So when we can get close, that I feel is pretty impressive. So I don't think we need to have all of those pieces in order to really get an idea, though, of how it happened. We're missing details, absolutely. Like anything in science, we don't have all the answers. But as it says, we have some very important clues. So I'd like you to think about this image for a second. How did life start? Well, we're looking at this image. It's obviously a jigsaw puzzle, but it's incomplete. We don't have all the pieces there. However, we have enough of these pieces to know that we're looking at a photograph of oranges. We're not looking at lemons. We're not looking at tomatoes. We're not looking at an apple pie. It's oranges. Now, in the upper left corner, could there be a strawberry or a cherry in there? Sure, that's a possibility. But right now, with the number of pieces we have in place, we know that we're looking at oranges. And that's an analogy I like to use for how the current microbiological research is going on understanding the origins of life. We don't have all the puzzle pieces. However, we have enough of them where we're starting to see a picture. And we'll admit we don't have all the details, but we're starting to see a picture of how it happened. And that's enough of a picture to kind of rule out some other possibilities. Now, the understanding of the origins of life on Earth is not a new field of study, but I think some of the most important work really started in the 1920s, and as we've done so far in the class with other things, I'd like to talk about some of these pioneers, that even though they may not have gotten everything right, but still it's upon their shoulders that we're standing on to get the next branch of knowledge. Alexander Oparin in 1924 was really imperative and very, very important in the understanding of early life. So Oparin really started getting the ball rolling on what that early Hadean or late Hadean to early Archean world would have been like, and how could life have started. So Oparin pointed out, based on our geologic knowledge of the late Hadean, early Archean, the oceans and seas were hot or very warm, not cold and pretty frigid like today. Okay, fair enough. He also noted that based on geochemical evidence of Archean rocks, early atmosphere would have had lots of ammonia, methane, hydrogen gas, and also water vapor in it. Again, well supported, as well as some other things, but this is very well supported. Energy and lightning and solar radiation would have caused gases to combine and precipitate into the warm seas. Okay, so you add an extra source of energy to these gases that we have in the atmosphere, and you will get a chemical reaction. Well, we certainly know that today, if you take certain chemical compounds and you add energy in the form of electricity or radiation to them, they will behave differently and sometimes combine and will then change state as well, perhaps into a precipitant. So, fair enough as well. Gases combine, increase in complexity, and formed life, and then biotic evolution progressed. Now, that's a leap right there. Right about there. We're missing quite a few steps there, because he went from combining gases into precipitants, in other words, taking molecules and making them more complex molecules, to formed life. So let's dive a little bit more into that, because I think Oparin was definitely on the right track, but he was taking baby steps and then decided to pole vault to the next stage. So we should back up a little bit and look at that, because he's missing some very important steps. How do you get life to form from combining gases? It's not that simple. Well, one of the main things, though, that he's talking about with that energy combining and getting these compounds is complex organic molecules. Again, long, complicated chains of oxygens, hydrogens, and carbons that really make up all life on the planet. How do you get those? Turns out they're not that hard to find. Complex organic molecules can form abiotically, meaning you don't need a living thing to necessarily produce them. Turns out these are also some of the things that are formed inside of stars when they're undergoing nucleosynthesis. So stellar nucleosynthesis, when we're making carbon through iron. Remember we talked about the elements that we're producing during the various stages of nucleosynthesis. This is when stars are burning through their hydrogen fuel before they go supernova. They sometimes also make complex organic molecules. They will bond together carbons, hydrogens, and oxygens. Molecular clouds in space, from when those stars do happen to go supernova, they will cast those gases, those complex organic molecules out into space, and they'll form in nebular clouds. So they're floating around in space. They're not hard to find. They also will bond together in our atmosphere. In our atmosphere, we have free gases of carbon dioxide. Those are your carbon and your oxygen. We also have water vapor, hydrogen, and oxygen. We also have hydrogen gas, ozone. All of those can bond together with solar radiation and lightning and produce complex organic molecules. So in other words, the basic ingredients that you need for life, the nuts, bolts, screws, and nails, and the glue that you need for life, are all around us, and they're all over the solar system and all over the universe. So we already had the Lego pieces in place. We just needed something to assemble them and really get them moving. And I wanted to point out too that the carbon, we're all carbon-based life forms, whether you're a bacterium or a chimpanzee or a human being or a dinosaur, you're all carbon-based life forms. And a lot of that carbon that we have on Earth actually came from meteorite bombardment from the Hadean through the Archean. So the carbon was getting here usually in the form of these complex organic molecule chains. So this stuff is around, but today we don't have a planet that is the same conditions where those complex organic molecules could start the process of potentially forming life. Too many things have changed. So we try to go back and look at the Hadean, look at the Archean, and figure out what exactly was going on at that time. So I want to talk a little bit about the Miller-Urey experiment. In the 20th century, 1960s, 1950s, 1960s, Stanley Miller was a graduate student at the University of Chicago, and Professor Urey was his PhD advisor. And Miller really wanted to test Oparin's hypothesis about if you take gases and you zap them, will you produce life? And the way that graduate school commonly works, Miller brought up this idea to his advisor. Urey said, yeah, whatever, try it out. He tried it out. They got some very interesting results, and of course, Urey wanted to be on the manuscript and be published in the paper after that. So that's why it's known as the Miller-Urey experiment, even though it was mostly Stanley Miller. So what they did is, my highlighter here, they started with this system where it was a closed system of glassware, and in the bottom they had a sterile pond, essentially, this beaker full of water, and it was sterile, and they're going to use that to replicate their ocean. Okay, and they have this little device, a stopcock, so they can pull water out and test it, and they're, yep, it's pure H2O. And then as we go up the chain here, or up the tube, they have this other opening where they can add gases such as hydrogen, methane, and ammonia. And so they add those gases, they go up through this pipe down into this large chamber which has tungsten electrodes, and so essentially they would zap it. This sounds very Frankenstein, doesn't it? And so they had these gases going through, and they electrocuted them. They zapped them with tungsten electrodes. And then they had those gases pass through a condenser. So we would actually get precipitation similar to how a cloud forms. So they had gases, and then they cooled them off so they would become little rain clouds. And the moisture would then travel down through here and back. So again, they start with heated water that is going to drive up. So we have water vapor mixing with the ammonia, methane, and hydrogen gas, and it goes through the tube into the tungsten electrode. It gets electrocuted, and then it condenses in it, then returns back to the basin. And when they took their second water sample after doing this, they found it wasn't just water anymore, nor was it water with methane and ammonia and hydrogen in it. In fact, they had made amino acids. Amino acids, these essentially chains that can be built from simple organic molecules. Amino acids are the basis of your DNA, of all of the cells and molecules of your body. Amino acids make things work. So they had actually created amino acids. They did not create life like Oparin had said. However, he was correct enough that you, in doing this process, or in the Precambrian, in the Hadean, Archean, you could actually produce the raw materials that were then needed to produce life. Okay, so one of the most important steps, how do you make complex organic molecules? Not just that, you can make amino acids, a very specific set of those complex organic molecules that are imperative for life. So now we need to figure out a way to get these complex organic molecules, these amino acids, together and to congregate and actually then start going through the chemical reactions that might start producing an early life form. Okay. So the oldest hypothesis for where we would get life forming is this concept of the primordial soup. Essentially the idea that billions of years ago in the Precambrian, amino acids were just floating around in the warm seas, getting occasionally electrocuted by lightning and solar bombardment, and they would just bump into each other and form life. The problem is, we just talked a little bit about the formation of greenstone belts in the last lecture. Most of the planet in the Hadean and the Archean is ocean. Microcontinents are pretty small. Even by the time we actually get continents forming in the Proterozoic, even today, most of the surface of the world is ocean. So what are the odds of these little organic molecules bumping into each other? Pretty slim, right? It's Pretty slim. So it's very unlikely that randomly floating amino acids are going to bump into each other and produce life in the early ocean. So the primordial soup is not likely. It's too dilute. So we don't actually think that is the way. It makes for a fun story, and you'll see it in old texts, but no, we actually don't think that the primordial soup is where life formed. What about mid-ocean ridges? Now, this one's kind of ironic, because mid-ocean ridges, which remember the center of like the Atlantic Ocean where seafloor spreading is happening, it's a major plate boundary, divergent plate boundaries associated with plate tectonics. Mid-ocean ridges and some of the features of those were first, I don't want to say discovered, but explored by a researcher named Dr. Robert Ballard. So Ballard is actually a geoscientist. However, he's most famous, and you may already know this, for discovering the wreckage of the Titanic. So the guy who actually has done research on what's going on at the bottom of the ocean and has pioneered all of these fantastic and amazing concepts of what we know about the ocean floor today. He's known for finding a very, very old sunken ship. But I digress. Mid-ocean ridges. How is this possibly a place where life is forming? Well, if you go down to a mid-ocean ridge, it's not just a big gash in the middle of the ocean. It's actually a series of these vents, very tall vents, and we call these black smokers. And they're volcanic and constantly spewing out hot water, especially other gases like hydrogen sulfide. You would need a lot of this type of energy in the oceans for life to occur. So it's possible we had amino acids formed from something similar to what O'Perrin was hypothesizing, and the Miller-Urey experiment shows how it could have been done. But perhaps it's at mid-ocean ridges where these compounds are combining to produce the amino acids. Possibly. What's ironic about that is prior to Ballard going down and taking photographs like this one, we actually thought life could not exist at the bottom of the ocean until Ballard went down and found that these black smokers actually have organisms living on them. Tube worms and some crustaceans and some echinoderms actually live on mid-ocean ridge hydrothermal vents, on these black smokers. Now, if they go in the vent, they are destroyed. They're boiled alive. But they feed off of this hydrogen sulfide gas. They feed off of the nutrients and all these minerals that are coming out of those vents. And they actually sustained it. They just have very, very specific heat sensing mechanisms to know, get close, but don't get too close. So we know that not only is life down there, it's teeming with life, and we think that this is a very likely place that life may have initially formed, is at mid-ocean ridges. And then clay. Clay plays a role. Now, the clay we're talking about here is what you would find at the bottom of the ocean. So the rocks weather on the continent, they wash out to sea through rivers, and the clays are going to drift out the furthest because they're very, very fine-grained, and they can take thousands of years to get to finally settle at the bottom of the ocean. Now, if you look at a clay mineral under a microscope, or better yet, look at its chemical structure, they form these sheets very similar to micas, like muscovite and biotite. That's why clays are slippery. You're actually slipping and sliding little microscopic plates past one another. The reason they're also so slippery is because each of those sheets and plates can peel apart very easily, very similar to micas. You remember muscovite and biotite and chlorite, you could peel sheets off in one direction. They have one good plane of cleavage, and clays are the same way. So what gets those layers of clay to stick together is the charge. They're kind of have a magnetic, like a positive and a negative static charge that holds them together. So if you separate them, they have a charged surface. So think about like if you take a towel out of the dryer without putting a dryer sheet in, and it's got static on it. Now you've got clays draped down around mid-ocean ridges. You have mid-ocean ridges producing lots of heat and lots of energy and a lot of gas, and it's possibly in those spots that these compounds are combining and producing amino acids, and the amino acids then get attracted because of the static charge to these clay minerals that serves as a structural framework to allow for these amino acids to combine together and start undergoing chemical reactions that can potentially produce the earliest, simplest forms of life. So out of these three, primordial soup, mid-ocean ridges, and clay, primordial soup is actually the one we think is the least likely to play a role in the formation of life. Mid-ocean ridges and clay probably fit together in tandem in how life formed. So again, ironically, we think life probably started in deep oceans when we used to think life wasn't down there at all. We now think that's where it may have started. So let's talk a little bit about what kinds of life we're talking about. All life are, a set of these complex organic molecules. They're packaged in something like a cell, and they undergo chemical reactions and self-regulate and self-propagate or reproduce that way. So all living things are built of cells. All cells reproduce by dividing. Sometimes it's done sexually, where you mix some genetic material and get something new that will divide, or some organisms just clone themselves, and that's asexual reproduction. Probably not as fun. So the three domains of life are bacteria, archaea, and eukaryotes. We are eukaryotes. If you notice down here, we are one branch, right over there. We're one tiny branch off of eukaryota in between fungus and slime molds. So some of us more related than others. If you look at bacteria, this is where we start getting things like cyanobacteria, gram-positive, green non-sulfur bacteria. These are the things that you can get that can make you sick. They're also the things that live inside your stomach and help you digest. Their bacteria is very, very important. It's just some of them don't vibe real well with us. Archaea are some of the extreme, we can't really call them bacteria, but they're extreme forms of life. In fact, we think the earliest forms of life were probably Archaea, which is appropriate since they probably formed in the Archean. These are things like methanogens, halophiles, hyperthermophiles. These are the type of microorganisms that can live in hypersaline waters in Antarctica. They can live in ice. They can live at mid-ocean ridges. The sulfur oxidizers. These are the extreme microorganisms. So those are the three main domains. Now we usually will group bacteria and archaea together. These two groups share quite a lot in common, and so we will group bacteria and archaea into a group called prokaryotes. And eukarya or eukaryotes are their own branch off of that. So let's talk a bit about prokaryotes. Remember, bacteria and archaea. These are the smallest of the smallest life forms on the planet. Okay, so if you've ever seen anybody take a petri dish and they will, put their finger on it or something, and then a few days later you see, bacterial colonies growing over it. That's what we're talking about, these little prokaryotes. They're very, very primitive. They're all single-celled. All prokaryotes are single-celled organisms. Now, what's weird about them is sometimes they form big groups, and that big group will function as a single organism. And that's kind of creepy, but they do that. That's why some people, if they're bedridden for a long period of time and they develop bed sores and those get infected, that's because what's essentially formed is something called a biofilm, which is when prokaryotic organisms start acting like a common single organism, where they go through a whole bunch of changes and they are 100 times more resistant to antibiotics. So that's really kind of scary stuff. Biofilms are something that are quite common, though. I mean, if you brush your teeth every morning, you're removing biofilm from your teeth. Okay. So prokaryotes have been around, we know from direct fossil evidence, from at least 3.2 billion years ago to obviously very abundant today. Again, not to creep anybody out, but these things live in your hair. They live on your skin. They live in your skin. They live in your gut. I mean, if you've ever taken antibiotics and noticed that you've had digestive problems afterwards, that's because those antibiotics killed the important, happy bacteria that live in your stomach that help you digest food. So we have these living things living in us, and we actually kind of need them. So they're really around all over the place. And this includes cyanobacteria. Cyanobacteria is a very, very important type of bacteria, of prokaryote. So we have cyanobacteria today. This is what they look like here. You can see they're these little tiny squares that are stacked in these filaments, and they're very, very tiny. So you've all seen this before. If you've ever in the summertime looked at a pond or a lake and it was green on the surface, or if you had a swimming pool and you didn't treat it, congratulations, you're looking at cyanobacteria. It's pond scum, more or less. It's pond scum. Now, cyanobacteria have been around since the Archean, and we actually find fossils of them, both body fossils, sometimes in things like cherts. More importantly, we find their trace fossils in these rock structures called stromatolites. Now, stromatolites were very abundant in the late Archean, middle to late Archean. Today, they only live in two places, Antarctica and one reef in Australia. Essentially, how a stromatolite works is it's an algal mat, or a mat of bacteria that lives in the water. Now, these particular bacteria derive their energy and their living functions from the sun. They are photosynthetic, just like plants today. So they'll absorb solar radiation, they will breathe in carbon dioxide, and then they will exhale oxygen and produce sugar, glucose. Right? It's basic form and formula for and cycle for photosynthesis. Now, if you're living in a little mat in shallow water, you want to be in shallow water because that's where the sunlight can get through and you can start photosynthesizing. However, occasionally you get a storm event would come by and sediment would bury that mat. And that makes the cyanobacteria unhappy. So they grow new cells up through that sediment and they photosynthesize there until another round of sediment buries them. So stromatolites are these layered rock structures. The layers indicate the various layers of sediment, and then you'd get another round of bacteria, another round of sediment, another round of bacteria. And again, we still have these forming in a few places today. So they're little colonies. So the stromatolites are essentially where the cyanobacteria were living in. And there were more than just cyanobacteria living in them, but those are among some of the most abundant and most important ones. So stromatolites record that very first visible evidence of life, and they go back about 3.5 billion years ago, roughly. Now, the image you're looking at are a couple of stromatolites, a stromatolite reef from Sharks Bay, Australia. Okay, whether we're looking at the old ones or the new ones, there are rocks. They're just rocks, but they're produced accidentally from the colonies of these different kind of bacteria. And notice again, the ones we have today that live in shallow water, because cyanobacteria or blue-green algae is a very abundant form in those. So why do you think stromatolites today only form in environments where, in these very specific places, like certain places in Antarctica and certain places in Australia? Really, it's because nobody else wants to live there. There are a lot of organisms that would normally eat bacteria. They eat cyanobacteria, but not in these environments. So they live today in very, very harsh environments, so they don't get attacked. So they've found a good niche in there. Okay, so an important thing to remember about cyanobacteria or any photosynthetic organism is that oxygen is the waste product. They breathe in carbon dioxide, they exhale oxygen. And hopefully some of you are seeing where this is going. Okay. Plants do the same thing today, but they do it in a slightly different version. They've kind of figured out their own way of being photosynthetic. So, nature doesn't usually reinvent the wheel. There's some very strong relationships between how cyanobacteria photosynthesize and how plants do it, and there may be some kind of evolutionary record in doing that as well. So once again, here's a close-up of that stromatolite. You can actually see a little rock hammer here for scale. That's actually a regular-sized rock hammer. So these stromatolite reefs can be absolutely huge. This particular one is from the Wapame origin, and yeah, it's 1.6 billion years old. These things can be absolutely huge, and you're looking at millions of years of stromatolite production. Just like our modern-day coral reefs can literally build off of the remains of the old reef for millions of years, stromatolites were the first reef-forming organisms, but inadvertently. So here's Sharks Bay, Australia, where we actually find modern stromatolites forming. Most of the ones you're looking at here, though, are exposed during low tide, so those cyanobacteria get a little bit stressed. And these are hypersaline environments. These are environments where the salt content of the water is absolutely extreme. In Antarctica, where these hypersaline lagoons form, they're so salty and so cold, you stick your hand in there and pretty much instant frostbite. But they're living in these places, or they're colonizing these areas, and they're doing quite well because they really don't have any natural predators there. The cyanobacteria photosynthesize because nobody else wants to live in a hypersaline lagoon. That doesn't necessarily mean that it was like that in the Archean. There probably wasn't much else for life in the Archean, but our modern stromatolites live in these hostile environments. So, how does this fit in with what's going on in the Archean and the Proterozoic? Well, our stromatolites are going to be living in shallow water environments, on shallow continental shelves, places where the continent meets the oceanic crust in a shallow water environment. Why shallow water? Because they need that solar penetration through the water so they can photosynthesize. If you live in deep water as a stromatolite, you're going to starve to death. You need the sunlight. So they live in that shallow water. They're pulling in carbon dioxide, which is abundant in the atmosphere, thus it's diffusing into the surface waters, the shallow waters, and they're using that and the solar radiation, and they are pumping out their waste product, which happens to be oxygen. Now we have oxygenated waters. Well, also in that water is dissolved iron, especially Fe2, as well as some silica and some oxygen. Once that iron 2 oxidizes and becomes iron 3, and the silica oxidizes and becomes chert, it precipitates out, and we start seeing alternating bands of hematite and silica. And this should sound familiar because in the last lecture, we introduced banded iron formations. Now, banded iron formations tell us that in the late Archean, early Proterozoic, we started seeing a surplus of oxygen, but it didn't tell us where it came from. Stromatolites indicate the presence of cyanobacteria as the factory that produced the oxygen. The evidence for the oxygen are banded iron formations. So, important distinction to make. These are two very important rock structures that we see very abundantly in the early part of the Proterozoic to the late Archean that indicate how the atmosphere was changing. We see banded iron formations. What do those mean? Banded iron formations means you have oxygen in the atmosphere. Okay, cool. Where did that oxygen come from? Stromatolites provide that answer. So one is the cause and one is the effect. So you can see when we look back at our banded iron formations, the youngest ones we have are about 1.75 to close to 2 billion years old. And our youngest one, the oldest banded iron formations we have are around 2.5. So you can see here where we start out with very, very low amounts of oxygen in the Archean. Once we hit the Proterozoic, when stromatolites become abundant, oxygen skyrockets. So why don't we have abundant oxygen in the Archean? Why only in the Proterozoic? Well, remember, in the Archean, we're building continents. We're having all these collisions with the microcontinents. We're producing the greenstone belts, but most of that's happening through the late Archean. Remember also, Archean sedimentology is predominantly greywackies, indicating that you have rocks weathering off of continents and immediately becoming deposited without a lot of refining and recycling. That's not a good place to form a stromatolite. You have high sedimentation rates, and you don't have nice shallow water environments off of your coast. However, once we get the large continents in the Proterozoic, once collage tectonics starts slowing down, we have our continents roughly the size they are today, that's when we start seeing those shallow, passive continental margins, which are perfect places for stromatolites to grow. So in discussing greenstone belts and the construction of continents in the Precambrian, by the time we get to the Proterozoic, we have now produced an environment that is conducive for stromatolites to form. Once the stromatolites and cyanobacteria are happy and they're very productive, we see global oxygen concentrations go up in the form of abundant banded iron formation. So these three events go hand in hand. They just follow along. You build the continent, that's a place for the organisms start producing lots of oxygen, and you see that oxygen present in the banded iron formation. Okay. So around 2.4 to 2.2 billion years ago, atmospheric oxygen really increases. And here's another way of looking at it. Our cyanobacteria are in the shallow waters. They produce oxygen, which mixes with ferrous iron to produce ferric iron oxides, which precipitate as banded iron formation. Again, we don't have this forming very much in the Archean, middle or early Archean. We start seeing it in the late Archean, and it really takes off in the Proterozoic, because that's when we have a stage set for these stromatolites to form. But remember, stromatolites are stuck in place, and they're producing oxygen. We don't yet have a lot of organisms that are producing carbon dioxide. In other words, we don't have a lot of respirating organisms, because we don't have enough oxygen yet to become a surplus. We're getting there, but it's not quite yet at this point. At this point, photosynthesis is still one of the more abundant forms of, of breathing and metabolizing. There are other sulfur-reducing bacteria and other things around too that don't breathe oxygen, don't breathe carbon dioxide, but other things as well. So banded iron formations are really abundant. Pretty much if we look back at this 24-hour clock of Earth history, they are forming from about 6 in the morning till 1:52 in the afternoon. That is the large chunk of the Precambrian, or of the Earth's history, I should say, where we're just building up oxygen. We're just building up more and more oxygen. And then all of a sudden they stop forming. We don't get banded iron formation anymore today. We still have oxygen in our atmosphere. We have plenty of things photosynthesizing, but we don't find banded iron formation forming today like we did back in the Precambrian. So why is this? What happened? Once we start building up enough oxygen in the atmosphere, we start seeing the evolution of single-celled organisms. We're still dealing predominantly with prokaryotes that are going to start being aerobic. The bacteria that do breathe oxygen and exhale CO2. These start to become more abundant because now there's this new resource in the atmosphere, oxygen gas, that they can use, and it's very efficient. And among these early oxygen-breathing organisms, we have plenty of prokaryotes doing it, but that's when we get our first true eukaryotes. More complex single-celled organisms, and eventually multi-celled organisms. So what is a eukaryote? Well, eukaryotes include amoebas, they include all plants, fungi, as well as animals. So we are eukaryotes. And you'll remember that prokaryotes were just kind of a simple cell. Eukaryotes are much more complex. They have things inside of their cells that do different things. They have a nucleus, which prokaryotes don't. They have mitochondria, Golgi apparatus, and all of these other things that perform functions for that single-celled organism or multi-celled organism in the cell. Okay, they are much larger than prokaryotes. In fact, if you look here at these little mitochondria and ribosomes, riboclast, and nuclei and stuff, and Golgi apparatus, all of these organelles that are inside of a eukaryotic cell, each one of those is about the size of a prokaryote. So eukaryotic cells are a magnitude larger than prokaryotes. The DNA is located in that nucleus. It's isolated. It's not just in a nuclear area like we saw with prokaryotes, which are very simple. These have a very specific place for DNA. The other thing that's interesting is that eukaryotes, or many eukaryotes, not all, but many eukaryotes, allow for sexual reproduction, which means the mixing of genetic material. Now, if you have a single-celled prokaryote like a bacteria and it reproduces, it just divides itself. So it's essentially cloning itself. With eukaryotes and sexual reproduction, you're actually mixing genes from column A and column B, and you're getting something brand new. So once we get eukaryotes on the scene, evolution starts going into overdrive compared to what it was in the earlier parts of the Precambrian. The oldest eukaryotes that we know of are these little algal organisms, algae, called Acritarchs, and they first appear about 1.75 billion years ago. And what's really important about eukaryotes is they are photosynthetic. They're an algae, so they're kind of like a little single-celled plant. But they're coastal plankton, meaning they're floating in the surface waters. They're not like stromatolites or cyanobacteria where they're anchored down to the bottom of shallow water. These things actually free float on currents. So they start out as coastal plankton, and eventually they start covering the waters all over the world. It's like if you go to the beach and they say you can't go in because there's an algal bloom or red tide. It's things like this. Okay. Today we find them as fossils. We have to etch them out of rock, and essentially these little spore-like sacs that they would grow around themselves. So this guy right here, what you're seeing with this crescent shape, is actually the tear where the reproducing or growing Acritarch shed that and then grew another one. So it's almost like a trace fossil. But with Acritarchs came quite a lot of change, further change on the planet. Because remember, these are algae. Algae photosynthesize like other kinds of plants. So the first appearance and then diversification of Acritarchs is a huge event in Earth history. The first eukaryotes, Acritarchs, indicate this huge leap in global productivity. We see a huge increase in photosynthesis because now it doesn't matter how deep the water is, they're only living in the surface. So they're photosynthesizing all day. And with that, we see an even bigger, huge leap into atmospheric oxygen. So you remember we said in the last lecture that it wasn't until the Proterozoic that we actually got oxygen to about half of what it is today. That's thanks to Acritarchs. So this particular group of single-celled organisms almost single-handedly changed the way that our atmosphere works. Not only that, but about 1.75 billion years ago, something much more important happened with Acritarchs. And that is, Acritarchs invented sex. So what's so great about sex anyway? And I'm really glad we're doing this as kind of a podcast because that's a rhetorical question. The big thing is it allows for mixing and differentiation of genetic material. Okay, Acritarchs didn't clone themselves. You know, one Acritarch would bump into another Acritarch and they'd chat each other up and have a baby Acritarch that was a little bit like mom, a little bit like dad, but something totally unique. And just like we talked about when we discussed the theory of evolution and we discussed natural selection, that variation drives evolution. Variation is the matrix of evolution. So it's those kind of new additions that we see that really kick evolution into overdrive. It makes a more rapid evolution possible. We're still not going to see a new species with every generation or something like that. But compared to what we were seeing before, it's considerably faster. So these things diversify throughout the later Proterozoic. They continue to photosynthesize. And it's also around this time we start seeing oxygen-breathing eukaryotes as well. But Acritarchs are really, really important for those two things, increasing global productivity and also really kickstarting sexual reproduction, which then drives evolution much faster than asexual reproduction. So we do have a couple of things, though, we need to think about. First, how do we go from a prokaryotic cell, a simple prokaryotic cell, to a eukaryotic cell, which is much more complex, it's larger, and it can do more things? How do we go from one to the other? We didn't really talk about that. Also, how do we go from a single-celled eukaryote, like an Acritarch, to a multicellular metazoan, which is any multicellular eukaryotic organism? Turns out both of these questions could be answered by the same thing, and that is a process called endosymbiosis. And I've already kind of alluded to this a little bit. We mentioned before that you have bacteria that live inside of you, right? And that may creep you out, but they help you. You give them a place to live, and they help you break down things like vegetables, okay, and help you ferment those and help you digest them. Cows are a great example. You can always tell when you're driving by a dairy farm because of the smell, the methane. Methane is a gas that is released by a certain type of bacteria that live in the guts of cows. Cows eat vegetation all day. It's very, very hard to break down. Cellulose is plant material is very, very hard to digest. So cows have these bacteria living inside of them, and it helps break that down. And one of the byproducts is the methane gas that they emit, but it helps them break down those complex sugars and also the hard cell tissue. So symbiosis is a phenomenon with organisms where you have two organisms that mutually benefit each other. This isn't parasitism, where one organism is benefiting off of the destruction and harm of another. No, in this case, both are benefiting. So if you look at sea anemones and clownfish, yes, Finding Nemo, that is a symbiotic relationship. The clownfish gets a place to live that's protected because sea anemones are venomous, and the sea anemone gets a clownfish that cleans the algae off of it. So they both win. Sharks and remoras. Remoras are these little like sucker fish that hang on the back of a shark. The remora is happy because it's living on a shark and nobody's going to mess with it. And the shark's happy because the remora will eat parasites off of it. So symbiotic relationships are very, very important. Endosymbiosis is a symbiotic relationship where one organism is inside the other and it's protected and given nutrients, but it also benefits both parties. Okay? And that's what we think is happening to produce not only eukaryotic cells from prokaryotic cells, but also single-celled eukaryotes to multicellular life. So the way this works, and this is from figure 9.10 in your textbook, if you look at plant cells, the chloroplast inside of a plant cell, the thing that helps it photosynthesize, is the same size, same shape, and same structure as a cyanobacteria. If you look at an animal cell, a eukaryotic animal cell, there's a mitochondria. Some of them have little flagella so they can move around. All of these things are actually single-celled organisms from the prokaryotic world that have been ingested by a larger cell, but wasn't digested. It was kept alive. And so they both function. And if you ever look at how especially animal cells reproduce, undergo cellular mitosis, everything divides at the same time. There's a quorum sensing. It's the signals that these organelles send out saying, hey, it's time to split, and everything splits in sync. So it's single-celled organisms functioning as a common organism. So endosymbiosis, we see a lot of modern-day endosymbiotic relationships when we look at our own cells. It's very, very common. It could be the similarities between what we, our bacteria, the little eubacteria, the prokaryotic organisms, the organelles, the structures inside of eukaryotes are very, very striking. They're the same size. They're the same function. They reproduce the same way. So we really think endosymbiosis is probably the key to how we go from prokaryote to eukaryote, and in many ways how we go from single-celled eukaryote to multicellular life, where lots of cells are functioning as a single organism. And so this kind of concludes the content for Unit 2. And I want to draw your attention to page 200 and 201 in your textbooks. You see this two-page spread, and it talks about really from the origin of the Earth 4.5 billion years ago. And yes, I know we started this unit far before that with the Big Bang, but this is from 4.5 billion years ago to the Cambrian, about 550 million years ago. And we're just going to stop before that. I want you to focus on this one with the Hadean, the Archean, and the Proterozoic. Notice that the top band is atmospheric events, then we have biological events, and then we have geological events. And remember, the first day of class, we talked about the complex interaction of the Earth's system of life, atmosphere, geology, and so on. And that's what you're looking at here. Again, notice that in the Archean, after we start getting, we start building our greenstone belts, and we start, colliding continents to make larger continents. We have continental shelves. It's then in the latter part of the Archean early Proterozoic, we see the proliferation of stromatolites, because now they have a place to grow. And then shortly after that, if you look up at atmospheric, that's where we start seeing the banded iron formations and the increased abundance of oxygen in the atmosphere. So this shows the relationship of all of those. Now remember, you're going to want to kind of think before this, because we did talk a lot about the origins of the universe and the Big Bang and the formation of the solar system and the planet Earth and so on. But this is a good visual, I think, to kind of hang some of your notes on for the last couple of lectures. And just remember that you can expand it out for it. So along with the study guide and this and the notes, you should be set. So to help you prepare for exam 2, here's some information for you. The exam will be available from noon on the 19th of March until 11:59 p.m. on the 22nd. So you'll have half the day on that Friday the 19th, all day Saturday, all day Sunday, all day Monday. You will have 90 minutes to complete the exam. Once you start, you have 90 minutes. I have posted a study guide to Canvas. It's an outline of the notes, plus you have the notes and you can review all the lectures. And like I did last time, I will be holding review sessions. I'll be doing a virtual one on the 17th, so the Wednesday before the exam, from 9:10 to 10:10 a.m. on Collaborate Ultra. And on the 19th from 9:10 to 10:10, the Friday the exam launches, just a little bit before the exam launches, will be a live in-person review session in Halsey 107. I encourage you to join us for one or both of these, however you can and feel safely doing. Please practice your essay responses, which are: be familiar with the Big Bang Theory, the different types of nucleosynthesis, and the nebular hypotheses in relation to the development of the solar system. So pretty much that first main lecture we did. Be able to discuss the process of continental accretion and the formation of greenstone belts. And what does this tell us about how continents are formed during the Precambrian? Discuss the oxygenation of the early Earth's atmosphere, including evidence and byproducts in the geological record. What's the difference in a banded iron formation and a stromatolite and a cyanobacteria? And finally, put these three concepts together. Demonstrate an understanding of the relationship between tectonic events, biological events, and atmospheric events that have shaped our planet in the Precambrian. After this exam and after spring break, we will start with the Paleozoic era, so the beginning of the Phanerozoic, and we'll continue our trek through the Earth's history. So until then, good luck, and feel free to email me with any questions.